Good afternoon or good morning, everyone. Thank you for attending this webinar. My name is Bruno Charletti. I'm the product manager at Fluigent, and I am today with Professor Jean-Louis Viovy, researcher at Curie Institute and co-founder of Fluigent. I am also joined by Dr. Alexandre Grassart, head of the research team of mechanobiology of host microbe interactions at INSERM. And we will be discussing all together flow control in microfluidics. So here's the presentation outline. I will first explain why flow control is generally needed in microfluidics. I will introduce the main liquid handling concepts to understand while performing microfluidic experiments. I will then cover the technology available for microfluidic flow control, how they work with their respective advantages and limitations. Professor Jean-Louis Viovy, pioneer in microfluidics and pressure-based flow control, will explain how the reason behind the creation of pressure controllers in his laboratory more than 15 years ago. Next, I will illustrate the performance of some flow controller in a typical microfluidic application, cell culture under flow. And finally, Dr. Alexandre Grassart will provide an in-depth discussion into the use of flow controller with the specific case studies in organs on a chip. Okay, so microfluidic flow control is a crucial part of your system as it will impact the results and the reliability of your microfluidic applications. So let's take the example of droplet microfluidics. Droplet production using this kind of system was implemented for application where the monodispersity is highly important. So as example with digital PCR or for cell encapsulation. So as you can see on the left picture, flow rate has a direct impact on the size of the generated droplets. So having a great control over the flow rate will ensure that you have consistent droplet size. Using microfluidic technology for cell culture is also interesting. So in fact, it offers many benefits compared to the traditional methods such as Petri dishes or well plates, as it will provide better, it will better reproduce the physiological conditions. So one crucial aspect of this technology is the type of the perfusion system you will be using. So in fact, unstable flow patterns will not mimic in vivo conditions, and it can also damage cells due to the uncontrolled shear stresses. So finally, it's also important to precisely monitor flow rates for fluid automation, are also for some unpredicted events, such as bubble formation or clogging. So for those reasons, it's important to understand microfluidics flow phenomena and select the right flow controller. So most microflow properties are derived from fluid mechanics. The specific conditions of microfluidics, such as the small dimensions and the micro to nanoliter volumes, will lead to very specific properties and phenomena. So amongst them, two main phenomena are related to flow control. The first one is laminar flows, and the second one is the hydrodynamic resistance. So those phenomena, in our experience, should be understood by microfluidic users. And so during this webinar, we will be focusing on them as they are related to the microfluidic setup and the flow controller in use. So let's have a look on laminar flows. So laminar flow, it's a type of flow regime or a fluid where the fluid travels smoothly in regular paths, as you can see on the picture. So it can be put in contrast with turbulent flow, which is characterized by chaotic, irregular changes in pressure and flow velocity. So laminar flow, it's a key principle in many microfluidic applications, such as in biological and chemical applications, where, for instance, in biology, you need to apply a precise shear stress to the system, or for controlling mixing reactions in chemistry. So there is an equation that allows you to accurately predict if you're under laminar or turbulent flow. This is the Reynolds number. It's derived from fluid mechanics equations, and it's a unitless number that indicates whether you are in the turbulent or laminar flows. So this number will depend on the fluid velocity, as you can see, fluid physical properties, as well as the system dimensions. So in general, for values of the Reynolds number less than 1000, the flow is considered to be laminar. For higher values, usually more than 1000, even 2000, it's considered to be turbulent. So if one wants to ensure laminar flows during an experiment, fluid velocity and the flow properties, including flow stability, must be well mastered. So now let's have a look at the hydrodynamic resistance. So it's also called flow resistance or just microfluidic resistance. So it corresponds to the opposition offered by a fluidic element. So in microfluidics, it can be tubing, sensors, and of course the microfluidic chip. So each element of a microfluidic system will offer some resistance to the flow, which translates into a pressure drop. So many of us often introduce it as with an electrical circuit analogy. So in fact, just like the electrical potential drop is proportional to intensity, a fluid pressure drop will be proportional to the mean flow rate. And in both cases, a resistance can be defined as the proportional coefficient. So again, like an electrical network, equivalent microfluidic resistance can be calculated to simplify modeling of microfluidic setup. So if you know how to calculate the total flow resistance of your system, depending on the controller you are using, you can have access either to the flow rate that needs to be applied to achieve a specific pressure, or the pressure that's required to achieve a specific flow rate. So how to calculate and adapt this microfluidic resistance? So as described here, the total system resistance is the sum of the resistance of the macrofluidic components. So for instance, here we have the resistance of the tubing, the resistance of the chip, the second tubing, and the sensor. Depending on the component geometry, different formula exist to calculate this resistance. So for instance, we have here the equation for cylinders as well as for rectangles. So you can observe that the resistance will linearly vary with the channel length and the viscosity of the liquid used. However, more importantly, I would like to draw your attention that the fact that the fluidic resistance will vary inversely with the channel size to the power of 4. So instead of increasing or decreasing tubing length, it can be very interesting to change the internal diameters of the tubing. So it will allow you to have way more drastic changes in resistance within your system. So I hope you find this section informative and that some of those concepts can be already applied to your research. So now we'll have a look on the flow control technologies for microfluidics. So first of all, let's see what are the main components of a microfluidic setup for those that are not aware of microfluidics. So the setup will consist, of course, of a microfluidic chip, then a liquid container, so a reservoir, a syringe, or a bottle. A flow controller can be connected to the container to push the liquids into the microfluidic chips using tubing. So flow or pressure sensor can also be added to monitor in live those physical values. Microfluidic valves can also be used for more complex protocol, such as automated injection or multiplexing. Finally, one can also use software for automation and a microscope for live monitoring. So in this webinar, of course, we will focus on the flow controllers. So there are many flow controllers available for microfluidics. We here focus on three main systems. Those are the main system cited in the literature. So first of all, we have the syringe pump. They were the first system to be used for microfluidics, and it's quite basic. A syringe is placed on the pump, and the piston is moved by a stepper motor. So it will allow to push the syringe and inject the fluids. So it exists a wide range of syringe pump, from low-cost syringe pump to more advanced ones. The second system is peristaltic pump. So with this technology, tubing is periodically compressed by rollers that will allow to move the fluid from the reservoir into the chip. So finally, there are also a microfluidic pressure controller, a technology that was developed by Fluigent more than 10 years ago. Simply, a pressure is applied to a sealed reservoir, allowing to push the fluids. I will now give the floor to Professor Jean-Louis Viovy that will explain way better than me how and why they had the idea of developing this disruptive technology for microfluidics.
Hello, so I'll share my screen. Okay.
Perfect.
Okay, so yes, after this nice presentation, or at least in the rather as a complement to the presentation, I'll bring a little bit of history on how this, what was our motivation, and how the whole story started. So deliberately, I show you a slide which is kind of vintage, because it's the head slide we proposed to innovation agency before starting Fluigent to get support and finding and indicating the team and the different application we had at this time. To go more into what has motivated us to changing the paradigm of flow control, it was a time of there was a lot of excitement about genomics, but at this time we didn't have NGS yet, and so most of DNA analysis was done by electrophoresis, which was, as compared to what we can do now, extremely tedious and expensive. To give you an idea, the first human genome did cost about was really a worldwide initiative, and it cost about 1 billion euros, and now you can have a genome with typically 1,000 euros or less. So we participated in developments to try to make this easier, faster, and cheaper, but still within electrophoresis. And especially we had developed with postdoc at this time, Patrick Doyle, who is now a brilliant professor at MIT. And we had the idea of separating DNA by replacing gels, in which electrophoresis was done usually, by arrays of microfabricated pillars in microfluidic chips. And you have example on the left. This is a microscopy with labeled DNA, which appear in white. And you see a time sequence of a DNA going from right to left, encountering a pillar, and the DNA gets blocked by the pillar, but it stretches and finally slips around. But this slipping around is depending on the size of the DNA, and this way DNA can be separated by side. And now if you see on the right an electrophoregram, you see that for different voltage, you will have, in this case, it was a system in which we had two different types of DNA molecule, and you see that two peaks appear. And so that was used at that time for DNA mapping, which was a necessary preliminary before doing sequencing. So this was a strong improvement at this time, because we had a separation time which was about 2 minutes instead of 2 days in conventional gel electrophoresis. But what was surprising was that in order to reach stable immobile liquid, the preparation time took at least 1 hour. And the reason for that was that the equilibration time and the time constant of flow in this system was extremely low. So if I go to the different technology for flow control we had at this time, and it was already, I will go fast because it was already presented by Bruno. All these different system, except the microfabricated pumps, but which has other drawbacks, on which I think Bruno will come again later on. These systems we had at this time had very long time constants. And the reason is also something which was already mentioned to you as we understood it at that time, shown by Bruno, is the fact that flow resistance depend with a strong power to dimension, and when you miniaturize, you have extremely high flow resistance. And if you take the electric equivalent that Bruno showed you, you have the equivalent of an electric circuit in which we have pressure, which plays the role of a potential, flow rate, which plays the role of the current, and flow resistance, which plays the role of resistance. But you have another analogy, which is a bit more indirect but still understandable, is the fact that in microfluidic systems, you have residual elasticity, which can be in the chips. For instance, PDMS chips are an elastic material. Very often you use tubings which are flexible and which also have some elasticity. And beside this, you have external volumes, like the volume of the tubing, volume of the reservoir, and so on, which are often high as compared to the volume of the microchannel itself. So this combination of residual elasticity and external volume has an equivalent in electricity, which would be capacitance. And if you have an electric circuit which have a high capacitance and high resistance, you have a system which has a very high time constant. Indeed, in some application, you can even use capacitance and high capacity. If you have low current, you can even use capacitance like a kind of energy storage system. And so this was really intrinsic, and we wanted to find a way to get rid of this problem. Essentially, on a very practical way, and without going to any equation, the idea behind that is that when you stop flow, for instance, you stop your syringe pump, you have additional volume which is stored in this elasticity of all the system, and this volume is very slow to evacuate when you stop the syringe because of the high resistance. And oppositely, when you start the syringe, you first build up additional liquid by deforming the system before it can go to the channel. And in addition, this can give some non-control of the pressure, which can damage your chip. And we found that using this electric elasticity, in general, in electricity, people prefer to do potential control of current by potential rather than control of a system by current. And this typically gives better time constant and more simple control, especially in electricity. When you have this problem of high flow resistance and high capacitance, you use a potential-based system and a divider bridge configuration in which these extra electrons are evacuated by a secondary source. So essentially what we did is a system in which we made a pneumatic equivalent of this divider bridge. And you see here what was our first prototypes, in which you can see, but of course in a much less miniaturized way than in current Fluigent systems, valves, pressure sensors, and calibrated leaks, which plays the role of external sources in divider bridges and allow to evacuate rapidly the extra volumes. And with that, we could obtain subsecond flow manipulation and reproducibility of sampling, which was completely impossible with other means. And you see, for example, an example on the left of two different DNA sampling curves. And you see even with this relatively primitive apparatus, the change of principle allowed to have something nicely reproducible. And the other thing we found with that, with regulation now based on a simple PID algorithm, we could get stability of flow, which is the red line here, and much better than what you can do with syringe pumps. Okay, so that was the beginning of Fluigent, because we had this for our own as you can see, very particular application, but we had when we had visitors of the lab and we showed them this, very often we had a strong interest, and people asked us, can we have your designs, can we copy it, and so on. So this happened 1 time, 2 times, 5 times, and so on, and we understood that en fait, indeed, the problem we had was very general, and it was absurd that everyone tried to reproduce these things we did, each one artisanally, let's say, and that it was better to industrialize it, make a product that would both save time and money to people. And of course, we also discovered in the process that by industrializing things, there was a possibility to continue improving and performance as much better than what we could do in the lab. And there were many innovations which were made by Fluigent in this on this basis, and also with contact with customers, Fluigent discovered that there were many additional components that people wanted to have, and the companies started to best serve customers. The company started to develop a line, as you will see on the Fluigent catalog or website, develop a line of many components to really give to customers whatever their application, to the best extent, all they would need for building all the building blocks for their microfluidic system. And also more recently, because the field of microfluidics extended strongly in biology and analytical sciences, and in particular being at the basis of new generation of analytical instruments and other application, especially in cell biology, as you will see with Alexandre presentation. Fluigent make a new step, focusing on application and avoiding users who are not microfluidic specialists, with system based on microfluidics using the Fluigent experience, but which can be used, which are kind of fully integrated bio or analytical or medical instruments, either being full microfluidic instrument developers. And you will see a nice example of that too. And with that, I stop, and thank you for your attention. And with just a slide, thanking our main financial contributors, and as well as the collaborators and people who helped us progress in this domain. So thank you very much. And of course, I'll be ready to answer questions, but I think this will be rather at the end of the webinar.
Okay, so to close this section on flow controllers, I wanted to end up with a summary of the advantages and the limitations of all those technology available. So I won't go into the details of all those technology, but for instance, let's take the example of syringe pump. As Jean-Louis introduced it, it's quite easy to implement. You have great average to low stability and also a low response time. With a peristatic pump, it's again also easy to implement. You can perform fluid recirculation very easily, but it also provides high oscillations or low accuracy. So using pressure control, you will get the best performance. So in terms of stability and response time, you will have access to the pressure and the flow rate that you can also control depending on the technology you are using. It's also convenient for sterile environments as well as to couple it with a bottle, for instance. And the limitations are that you need to couple it with a flow sensor. So it's in the fluidic line. And in addition, if you don't have a pressure supply at your laboratory, you will need to get an external pump. So a pressure pump, a flow sensor, a pressure controller. So that leads to eventually higher cost as well as to higher time of implementation. It's also worth mentioning the miniature pumps that are quite emerging technologies. So they are compact, lightweight, great performance, but I will not qualify them as smart devices compared to the previous technology, as it needs further integration and development. Of course, the performance are also degraded. So now that you have a basic understanding of the different liquid controllers and that we are all on the same page, let's see how they perform on a typical microfluidic application. So we will here discuss cell culture under flow. So for this experiment, we will perfuse cells that were previously cultured into a 2D microfluidic chip. Fluid recirculation was set for 24 hours using 50 microliter per minute. So for this recirculation, two kinds of technology were used. The first one, it's the peristatic pump, which is quite straightforward to implement. Then the second technology is using pressure controllers, in which we connect flow sensors and microfluidic valve to reproduce a fluid recirculation. So that was what we were doing before, but now Fluigent today provides OMI that will allow to perform the same protocol using one single device that will integrate all the required technologies. So the pressure source, the pressure control, the flow sensor, and the valve actuation to reproduce recirculation. So let's have a look at the results. So as you can observe at the beginning, before implementing flow rates and the cells are well adhered with already high cell confluency. So they are well spread, which suggests a great cell viability. So the cultures are now connected to the pumps and the flow rate is implemented. So we can observe about 40% variation around the same points using the peristatic pump. And in comparison, we observe about 2% flow rate variation using pressure. So after 24 hours of fluid recirculation, one can observe that perfusing the cells with such a level of variation will affect cell viability. So in fact, we can observe about 30% cell detachment due to these induced shear stresses applied on the culture. And in addition, if you can observe, the remaining cells have a round shape, which indicates a loss of cell viability or even dead cells. So when using stable flow rates, we can, however, the same level of cell confluency than initially and well-spread cells. So it indicates that a stable flow rate will ensure cell viability, and it confirms that that flow rate stability and accuracy should not be underestimated for cell culture experiments under flow applications. So now I give the floor to Dr. Alexandre Grassart, our second speaker, a researcher at INSERM that will present a case study on organs on a chip on microfluidics.
Okay, so thank you. I will share the screen. Okay. Is it okay?
Yes, perfect.
Okay. So good afternoon, everyone. So I'm Alexandre Grassart. I'm the head of the team MOMI at the Institut Pasteur of Lille. So today I will talk mostly on how we do organ chip, more precisely on gut on chip system. So Maybe as a start, I will define a little bit how we define how we understand organ on chip. And so for this, I will refer to the definition of the European Society for Organ on Chip, which say that it's a fit for purpose microfluidic device that contain living engineering organ substructures in a controlled microenvironment, and that you can recapitulate at least one feature of an organ, the dynamics, the functionality, and the response as observed in vivo, and with capacity to do it, to monitor it in real time. So basically you have three major components. One is the microfluidic interface. The second one is the cell culture chamber where you can grow your organoids or tissue. And the third interface might include biosensor or offer opportunities to observe by microscopy the tissue that is grown within these chips. So, well, there's some bugs. Sorry about that. So nowadays there is almost all organ that can be mimicked using these systems. And one of the main advantages is that you can interconnect because every organ is together, or at least two. And so you can start to recreate this physiology that is happening in vivo. What that was impossible to do with a conventional in vitro system. And so there's this idea of a human on chip or body on chip, with the most, let's say, dream is to better predict this pharmacokinetics and toxicity. So these systems are actually placed by many above the small animal models, because you can detect and analyze and responses with human physiology that is not working with small animal models, but you still preserve the in vitro capacity. So it basically it's filling the gap between the best, offering the best world of these in vitro and in vivo models. So you have, of course, the possibility to cross with other technologies, stem cells, organoid, and you have many applications, I say. And today I will just focus on one particular application, which is the disease modeling. And so more precisely, I'll mostly talk about our application to better understand infection process. And some pathogens actually have some very specific reservoir. They can be very restricted to some species. So as you know, for example, COVID, SARS-CoV-2 was initially restricted to bats, and after transfer to other models. So that's one of the key issues when you want to study viral infection, for example, because they can only infect certain models. That's the case actually for one bacteria that we are very interested in, this bacteria of high concern. It's called Shigella, that you can see here on the left, the red bacille. And it's high concern because you have we observed among isolates, clinical isolate, that there is a huge resistance, increasing resistance for antibiotics since the last 5 years. And nowadays it's actually classified in the top 10 priority by WHO. So that's what we are studying. But for that, you need actual good models to better understand how it's working and to test also new therapeutics. So you need to have a good model of the intestine, of human intestine. So you cannot basically use mice, it's not working. And that's what we are doing. So in the intestine, basically, if you are not very familiar, it's looking a little bit like this, and you have several layers of defense. The first one is the microbiota that could play a role. Of course, you have the epithelium that is well known, but you have also a layer of mucus and also the immune system. And all these components interact dynamically to control all this exchange to educate the immune system and trigger the good response against pathogen. So you need an in vitro system that has this ability to have all these in one. So gut-on-chip models are very important because they offer the possibility to do so. And there's actually different technologies that are defined as gut-on-chip. I won't detail all of them, but I recommend to read this recent review that described the gut-on-chip models. And I will focus my talk on this particular model that has been proposed some years ago by the group of Dornenberg at the Harvard University and the Wyss Institute. That is basically a microfluidic channels separated by a porous membrane. So you have two microfluidic channels. The porous membrane support the cell growth, and you can have different interface with the endothelial cells or immune system, and you can also have co-culture of microbiota or pathogens. And one of the key elements is that you can also introduce some biomechanical component into this system. And so one of the first mechanical stimuli that is important for the gut physiology is the peristaltis, which is due to the muscle layers that surround this intestinal tissue that will compress or stretch the tissue at certain strain and frequency, and that's what you can read on the table on the right. Some chips can actually allow you to test these factors. So one of the chip that you can see that is from one supplier is composed of these two channels. One is blue, one is pink, and you have on the side two vacuum chambers. And so by applying this pressure or this vacuum, you can compress or stretch your tissue. And so for that, you can use this pressure controller that is very precise, and that's what I will show you right here, where it's very accurate and fast. So you can really obtain very reproducible data when we want to calibrate your system. And so that we can see here, where you have the linear curve, and on the right you have the imaging of a chip without the cells, or with the cells, where you can see the stretching. And with that, here is another result that we obtained with the two groups. We collaborated, the group of Nathalie Sauvenet and Emmanuel Labrilla, where you can see on the left, you have the stretching of your tissue, but you can, through mathematical modeling, detect the stress applied on this tissue. And so what they found is that actually you can read the result on the image on the right, where you have the heat map of the stress applied on this tissue, and also the event of infection, where you can see a nice correlation between high stress and activation of the bacteria to invade the tissue. So the second possibility and important things to mimic is the luminal flow that is happening into the intestinal lumen. So you have always a slow luminal flow that is exerted within this lumen, and this induces a very, very low shear stress. So you need a system that is very robust to have always this flow at the very low rate. And so this is the strength of this pressure controller, because it's very stable, and even at very low level of pressure. And the advantage is that you can also combine this with a flow meter just before your chip, so you can always control the flow that is applied, and you have a back loop to the system, so it always regulates and monitor the flow that is going into your chip. And the chip and all the system can be placed very easily into an incubator, so the cells remain very happy. So this is an example of what we can obtain when we have this very slow flow and cyclic stretching. So basically you seed your cells at D1 with some ECM coating before, and the cells will go into 3D differentiation after about a week of under these conditions. And so to give a better view of how the cells maturate is when you look at this picture from the scanning electron microscopy, when you are looking at from the top of your chip, like a eagle eye, and you can see that the cells mature very nicely with this macrovilli on the top of the enterocyte, which goes the macrovilli. And they also form this big structure that resemble to this villi and cryptic area of the tissue in vivo that you can see here. But most importantly, that's when you control these mechanical forces and this 3D maturation, what we observed that is very important for the effectivity of Shigella. So when you introduce Shigella into the lumen, after a couple of hours, you will see a big massive destruction of your tissue. So the bacteria is here in green. On top is the control condition. And what we observe is like, that's the very nice thing of this system of organ chip, is that you can have full control of the different parameters that is impossible to do using small animal models. You cannot stop peristaltism or luminal flow basically. And that's, you can do it here. And we observe that is very important factors as well as the 3D structures. Another key aspect of the gut physiology is to have a co-culture of macrobiota, and that's what we are doing now in the lab. So we are building our own chip that you can see here, and we are using other more complex system to introduce and control the diffusion and co-culture of these bacteria with the help of the L switch, which will be a valve and help the introduction of the bacteria into the intestinal lumen of the gut on chip. And this is what happens. So the design is a little bit different. It's a snake shape of the channel. And what you can see, you have a 3D maturation of the intestinal tissue over transillumination. And the red areas are actually probiotics that express RFP. So they look like red. And you can see that we managed to have a nice co-culture over a long time point here, just 48 hours. Demonstrating this capacity of this system. So what it's look like in real life, it's a little bit messy. If you look at these images, that's the reality for a biology lab frequently. So you have different systems. That's why you have different tubing. So you have 5 chips here under the hood, but when they are placed under the incubator, you have the system that is outside, and you just need to pilot this with the computer. But if you are not that much an expert into this, you can go and use now this new system that is offered by Fluigent called OMI, that is fully plug and play. So it's much easier and safer, especially when you are using pathogens, where you just have this reservoir that's just open and pipette up and down, and you can easily sample your system. And we worked with this system for a couple of weeks, and we were able to demonstrate, have the same result that what we have with our classical system that we built, where you have no flow, the cells remain flat, whatever the number of days of maturation. If you apply a flow with a very, very low shear stress, you have a 3D structuration of your intestinal tissue. Demonstrating that these all-in-one solutions are really, really adaptable to any chips, because here it was custom made. So with that, I would like to acknowledge the people who participated in the studies of this gut on chip, specifically my team, and also Elise Delannoy, who is working on this new version of gut on chip system in our lab. My previous collaboration, the group of Nathalie Sauvolet, Sami Goba, it's, well, sorry, that's my mouse. Sami Goban, Elisabeth Labruyère, and my current collaborators for new chips development, and also for the microbiota part, Catherine Daniel, and of course all my fundings. And I will be happy to answer to any questions during the question and answer time. Thank you very much.
Okay, so to end this webinar, I wanted to quickly introduce our company. So at Fluigent, we want to help researchers and industrial to be successful in application where fluid control at the microscale level is critical. So we provide thousands of flow controllers worldwide. So with a presence in the US through a subsidiary and with more than 10 distributors around the world. So our products have been widely used in top research laboratories for more than 10 years. And Fluigent pressure controllers are cited in thousands of research papers. So with that, Professor Viovy, Dr. Grassart, and I would like to thank you for your attention. So I hope you enjoyed this webinar and you learned some concepts. We will be more than happy to answer to your questions. So you can also contact us at contact@fluigent.com or at my personal email that you can find here. So thank you for your time.